All the king's men, microgliocytes. Microgliocytes make up about 10% of all the CNS glial cells. They are immunocompetent cells that form the immune system of the brain and spinal cord. In the intact, mature CNS, microglial cells are located in all parts of the brain, with their density being the highest in the hippocampus, olfactory brain, basal ganglia, and substantia nigra, and the lowest in the cerebellum and medulla oblongata. Under physiological conditions, they are characterized by small bodies and thin branched processes. Each cell has its own clearly defined territory, and the processes of adjacent microgliocytes do not touch each other. In gray matter, the processes extend in all directions. In white matter, they are usually oriented perpendicular to the bundles of nerve fibers. The described morphology of microgliocytes is characteristic of the intact CNS, and in this state they are called ramified or resting. But in reality, they are never resting. In the intact CNS, microgliocytes actively perform several important functions, about which I will tell later. Currently, there are several antigens for immunohistochemical detection of microgliocytes: F4/80, CD11B, IBA1. Electron microscopy reveals in microgliocytes relatively large, extended, and irregularly shaped nuclei. In the cytoplasm, forming a narrow rim around the nucleus, lysosomes and phagosomes predominate, and lipid or lipofuscin inclusions are often found. Microgliocytes are derived from precursors originating in the yolk sac. The development of microglia is best studied in mice and rats. However, the main stages of embryogenesis of microgliocytes in humans are similar to those in rodents. It was shown that the first erythromyeloid precursors of microgliocytes are c-KIT-positive cells. They develop into mature cells in which the F4/80 receptor and the macrophage colony-stimulating factor receptor are activated. These cells proliferate and enter the developing brain using specific matrix metalloproteinases. In the first days after birth, microgliocytes continue to migrate to the brain and spinal cord. However, later on, their transport from blood to the brain parenchyma practically ceases, and microglia exists in the form of a cell-renewing, stable population. Once in the CNS, microgliocytes for some time have an amoeboid appearance. Then they acquire the morphology of ramified cells. Under condition of an intact blood-brain barrier, microglia are not replenished with monocytes circulating in the blood, and differ from them and macrophages of other tissues by several markers and mRNA. Microglial cells constantly monitor their microenvironment with the help of their processes, having for this purpose numerous receptors and immune molecule recognition sites. Any violation of brain homeostasis causes rapid transformation of ramified microglia into an amoeboid form. This process is called microglial activation. At the first stage, ramified microgliocytes increase their size, retract processes that become fewer but thicker, change the expression of various enzymes and receptors, and begin to produce immune response molecules. Many microgliocytes begin to proliferate and their quantity multiplies. At the final stage of activation, microgliocytes become phagocytic or amoeboid cells that act as the professional innate phagocytes. They become motile and using amoeboid-like movements gather around the damaged site. The microgliocytes perform a phagocytic function not only in the mature brain, but also in CNS development when these cells remove apoptotic cells and extrasynapses. After the brain damage, the expression of the major histocompatibility complex II and co-stimulatory molecules CD40, CD80, and CD86 in the microgliocytes is upregulated, which is essential for interacting with T lymphocytes. Thus, the microgliocytes also play the role of antigen-presenting cells in CNS. Role in vasculogenesis. In the developing CNS, microglia connects and directs the newly formed vessels. It promotes their branching, and a decrease in the number of microgliocytes leads to a decrease in the number of branch points of blood vessels. It has been shown that microglia directs the developing vessels by secretion of soluble factors and not due to the establishment of direct contacts with vessels. Role in myelination and neurogenesis. The neonatal CNS contains a pool of microgliocytes with a CD11C positive phenotype. These cells also express genes for neuronal and glial survival, migration, and differentiation. They are the main source of IGF-1, and selective depletion of its production by these microglia leads to disruption of primary myelination. Thus, this type of neonatal microglia delivers the signals necessary for myelination and neurogenesis. Synaptic remodeling. Microgliocytes destroy the synaptic connections of healthy neurons that are in contact with damaged cells. This contributes to the redistribution of functions between the undamaged neurons, thus ensuring the plasticity of neural circuits. Trophic function. Microgliocytes secrete neurotrophic substances, cytokines, extracellular matrix proteins, and growth factors, which allows them to humorally affect other CNS cells. Microgliocytes are the only type of phagocytic cells in the CNS parenchyma. At the same time, macrophages of bone marrow origin are found in the brain and spinal cord. They include perivascular and meningeal macrophages and macrophages of the vascular plexuses. Perivascular macrophages are located between the blood vessels and the limiting membrane formed by the perivascular processes of astrocytes. These macrophages play an important role in the phagocytosis of dying cells, maintaining the structure and function of endotheliocytes, contribute to the stability of capillaries, and maintain the integrity of the blood-brain barrier. The location of perivascular macrophages allows them to monitor the state of both the interstitial fluid of the brain and the blood. Meningeal macrophages monitor cerebrospinal fluid and act as a barrier against the entry of infections into the CNS parenchyma. The macrophages of the vascular plexuses, together with the monocytes, dendritic and mast cells, located here, perform the functions of the immune defense of the CNS. Thank you for attention.